Hello everyone, I'm Yanying Yu, a PhD student in Lars Barker's group in Helmholtz Institute for RNA-based infection research in Germany. Here, I present you our work, Automated Interference of CRISPRi Guide Efficiency in Bacteria from Genome-wide Essentiality Screens. CRISPRi is one of the many extensions of CRISPR technology, mainly used in gene regulation, and utilizes a catalytically inactive Cas9 variant, dCas9. The dCas9 RNA complex is guided by guide RNA to bind to the target DNA sequence without introducing double-strand breaks. For a typical CRISPRi essentiality screen, multiple guide RNAs are designed for each gene. Plasmids containing guide RNA sequence are then transformed into target organism. Counts of each guide RNA are obtained by sequencing the extracted plasmid from the organism after a certain time. This count can represent the survival of the population transformed with respective guide RNA. The full change of the counts compared to the start time point shows the effects of the combination of knockdown fitness and the guide RNA efficiency. However, the current computational approaches for prediction of guide RNA efficiency are based on Cas9 system and eukaryotic data. Overfitting problem of these tools was also pointed out in previous study. Therefore, a dedicated tool for CRISPRi guide RNA design is needed. To develop such tool, we plan on developing a machine learning model. Firstly, we extended the canonical feature sets to 580 features, mainly including 4 types of features. Firstly, genomic features describing gene organizations, such as distance to stop codon and the number of downstream genes in the same operon. Secondly, transcriptomic features describing the gene expression level. Thirdly, thermodynamic features such as minimum free energy of hybridization of targeting DNA and guide RNA. Lastly, sequence feature including single nucleotide features of PAM and guide RNA sequence, and dinucleotide features of 30 nucleotide extended sequence. Of note, some of the features are gene-specific while some are guide-specific. We collected data from 2 CRISPRi genome-wide essentiality screens in E. coli, namely Wang and Rosette respectively. We only used the guide RNAs targeting essential genes to better extract the design rules for guide RNA. Correlative differences due to the differences in the methods in 2 screens are reflected in the distribution of the log-to-log change of guide RNAs. Using AutoScikit-learn, a toolkit of automated machine learning, we optimized a random forest regression model, resulting in Spearman correlation 0.7798 between predicted log-to-log change and experimental log-to-log change. Considering the distinct 2 peaks in log-to-log change distribution and the common need of gauging the usability of guide RNAs, we then split the guide RNAs into 2 groups using k-means clustering and transformed it into a classification problem. Using different log-to-log change as clustering thresholds, we calculated the ROC curve and resulted in OC value 0.9. Using SHARP, we managed to explain these black balls and understood the feature effect. However, 7 out of the 10 top features with highest prediction attributions are gene-specific, suggesting a more reliable prediction by segregating the confounding effect from gene-specific features. To segregate the gene-specific effects, we firstly trained a gene model with only gene-specific features, and then we calculated the residual log-to-log change by subtracting predicted log-to-log change by gene model from original log-to-log change, and use it as input to train a guide model with only guide-specific features. After certain iterations, we evaluated the 2 models by comparing the sum of the predicted log-to-log change of 2 models and the experimental log-to-log change. We observed early convergence of 2 models and static performance during iterations. Because of the known effect of targeting coding strand and the guide model highly rely on this one feature, remove coding strand feature to improve interpretability of other guide features regardless of the decrease of Spearman correlation and OC value. Using a bottom-up approach, we further reduced the guide feature set and resulted in Spearman correlation 0.7553 and OC value 0.87. From the reduced guide model, we can see that the top features include sequence features near the PAM region and also features distinct from CRISPR-Cas9 system. We then validated the model by silencing GFP in plasmid using flow cytometric-based assay to avoid confounding gene effects. It resulted in 0.76 Spearman correlation, marked as CRISPRi in the figure, outperforming other CRISPR-Cas9-based tools. To sum up, we did not only develop a predictive model for guide RNA depletion in E. coli, we also successfully separated the guide-specific effects from confounding gene-specific effects. Here, we present a new perspective of guide efficiency and an approach for better understanding of the mechanism of the CRISPRi system. Our model establishes a general framework for extracting rules for guide design from essentiality schemes for other CRISPR-based technologies. Thank you for your attention.